Hey, all right, so protein synthesis part 2, grab those notes, and this time I want you to grab a separate sheet of paper also, and maybe even some colored pencil. We're actually going to draw this out, so make sure you have a separate sheet of paper drawn on tomorrow in class. Let me get my sheet of paper ready. Okay, so first I want you to draw a big circle, that's your cell. Okay, and then you got your nucleus. DNA's in the inside of it, and that's chromatin form. Nucleolus, and then you have your ER. And I'm going to make my ribosome a little bit bigger just so we can see it. Okay, so take a second, and we have to label all this stuff. So you've got nucleus, and I'm actually going to... Okay, now everything else is in here too, the mitochondria and all that, but just for the picture's sake, to keep it neat, I'm not going to draw that today. Okay, so I'm going to move my DNA up a little bit too. Okay, so on the last set of notes, or the set actually before that, we talked about how DNA, which is right here, copies itself and makes DNA, and that's called DNA replication. So that's before mitosis, DNA makes DNA, and that's not what this video is about, so I'm not going to talk any more about that. This video is about how DNA makes RNA, okay, and we talked on the last video that was called transcription. So DNA makes mRNA, and that process is called transcription. Okay, then the RNA, once it's made, it goes out to the ribosome and it makes a protein. And that is called, so an mRNA makes the protein, that's called translation. Okay, so looking at this picture, just remember that the DNA is going to unzip, like helicase unzips it, just like before. The DNA is going to unzip, but this time it's going to make an RNA. So here I have blue, and I labeled it RNA. So the DNA makes the RNA. Let's see if I can write that better. There we go. So the DNA makes the RNA. That process is called transcription. And then the RNA, so the blue thing, is going to come out and go through the ribosome. Think about the ribosome as a hamburger bun. And it's going to go through the ribosome, and then out is going to pop a protein. So that blue thing right there would be a protein on the top. Okay, so here we go. Looking at today's notes now, somebody came up with this. And that was the George Beadle and Edward Tatum thing that said one gene, which is a segment of a piece of DNA, makes a protein. Okay, so looking at today's note, so this was like big picture. I want you to make sure you understand this. DNA makes RNA, the RNA goes out to the ribosome, and that makes the protein. And we're probably going to draw this several more times, but I just want to make sure that you see the big picture here. Okay, so looking at today's notes, there's 3 steps what's going to happen. And so to make, it says mRNA synthesis and modification. The making of mRNA is going to happen inside the nucleus, right? And we actually saw that. Look at the picture. Happening inside the nucleus. Remember, the nucleolus is like a copy machine because it's where the RNA is made. So we went over that several times with Unit 1. Okay, so in order to make the RNA, it's going to take 3 steps, and those steps are initiation, elongation, termination. Okay, so let me tell you just easy way to remember this. Initiation, that's when you start something. If you ever join a sorority or a fraternity in college, you have to go through initiation, which is like at the very beginning. Okay, when something is initiated, that means it's started. So initiation is the first one. Elongation, well, that's like when the RNA is actually being made. The RNA molecule is being elongated, it's being made. Termination, that means end, fall. If you ever get, hopefully you won't, but if you ever get terminated from a job, that means you got fired. Okay, so start with there's transcription. The DNA is going to unzip. You can see right here, it's going to make DNA template, makes the mRNA. Okay, just as a reminder, notice that the RNA has U's pairing with A instead of T. So RNA has uracil, not thymine. Okay, so you want to know that. Okay, so initiation, and that's at the very beginning, and luckily you don't have to know the name of all the enzymes that do this, but it says a protein called a transcription factor. Okay, so a protein called a transcription factor attaches to you can see it right here, the promoter sequence. And so here, I'll look at the second picture. Here's the transcription factor attached to the promoter region. Then additional transcription factors are going to be added. So luckily, you don't have to know all those. Thank goodness. So here's what you got to know. DNA, and I'm actually going to draw it. I think I can draw a little bit better than that, like more simple. So here's DNA. DNA is going to unzip. So there it is, unzipped. Now, in one place, it's going to be yellow. I'll do it on the bottom. The transcription factors are going to bind. So there's the transcription factors in yellow. Transcription factors bind. This is all of that yellow right there is called the transcription initiation complex. What is it doing? It's starting it. It's the enzymes that are starting the process of transcription. Transcription is the process being done. Initiation refers to the beginning process. Okay, now, next thing that's going to happen. RNA polymerase is going to build, and it's shown right here in this picture. RNA polymerase is going to go down DNA, and it's going to make RNA. And so, I'll make this color red. Okay, so now RNA polymerase is going to bind right there in red, and it's going to go all the way down the DNA. And when it does, like if it comes to a T on the DNA, it's going to build an A. It's going to make C paired with G, A paired with U. Okay, so that's your RNA. So the RNA polymerase, remember DNA polymerase makes DNA, so it's perfect sense that RNA polymerase makes RNA. It's going to bind to DNA, go down the DNA, and make the RNA. So it's really that simple. Elongation, RNA polymerase, it walks down it and reads it, almost like Braille. And it's going to make the RNA. So highlight for me RNA polymerase 2 on your notes. And that's what builds the DNA. Okay, cells can make multiple copies of RNA because the DNA is left intact. So then the DNA is going to zip back together. Let's go with termination next. There's elongation. You can see the pink being made. So that picture really shows it. The blue is DNA. DNA is unzipped. And then the pink is created. And see, look at the letters. Thymine and adenine pair together. Cytosine and guanine. So the RNA is just a cheap copy of the DNA. Okay, termination. Basically what happens is it gets to the end, like RNA polymerase gets to the end, and it falls off. It's pretty much that simple. Okay, so like we said, termination, the RNA polymerase basically falls off and it's over. It reaches the stop, which is red right here. It reaches the codon, which means stop. Remember that a codon is on the RNA, and it's 3 letters, and 3 nucleotides. So a codon is any 3 letters. So there's a start codon, and there's different codons that make the RNA. And so 3 nucleotides make a codon. And this particular codon says, stop, everybody fall off. Okay, so you want to know initiation, elongation, termination. Those are the 3 steps to transcription. Okay, after we've got our RNA made, so you can see it here. After we've got our RNA made, you got to change it. So this is post-transcription modification. After transcription, we have to change that RNA. Okay, so we have to make, here it is, it shows you there's the RNA. And the first thing you want to do, it says on the front end, modification of the RNA molecule. So on the 5' end, we're going to add a cap. And I always think about this cap as like a hard hat with a flashlight on it. And that's going to help it get to the ribosome. And so after we make our RNA, and I'm actually going to go back to this first picture. I actually wanted to show you this one from the last PowerPoint. Okay, so just look at this box. Just look at the eukaryotic cell. Once the DNA makes the RNA, which is right here, there's your RNA, you got to change it. So on one end, you're going to add a cap. So here I'm going to make my cap in red. Remember I said I think about it like a flashlight. So on the 5' end, you're going to add a cap. On the 3' end, you're going to add a tail. So you see my cap, you see my tail, and the tail helps it swim to the ribosome. And then you're going to have to cut out the stuff that's not important. And so if you look at this picture right here, it's showing you're removing the stuff that's not important. Now, the stuff that's not important is called introns. So I want you to write this in. Introns are junk DNA. Introns are junk DNA. And so you're removing the introns off the DNA. And so just the good stuff is called the exon, and you can see that word right there. Just the exons are going to go into the ribosome. Now, when I first learned that, I was like, I don't understand why is an intron, which means inside, and exon, which means outside. Well, think about it. We're still in the nucleus. The introns are going to stay in the nucleus. Introns stay in the nucleus. Exons do what? They're leaving the nucleus. Where are they going? They're going to the ribosome. So look at this picture. It kind of sums it all up. This is a great picture if you can pause it and try to understand this. Here's your DNA. DNA unzips, makes the pink RNA right there. So DNA unzips, RNA polymerase goes down, and RNA polymerase is even labeled. RNA polymerase goes down the DNA, makes RNA. Okay, good deal. Got that far. Now, we have to change that pink strip of RNA that's been made. We're going to add a cap, 5' cap. We're going to add a poly A tail. And we're going to remove, see how we're removing that? It's coming out. Snip out the introns. And so the introns will stay and the exons will go out. One thing I didn't mention that's in your notes, the spliceosomes, that's what snips them out. So think of spliceosomes as the enzymes that cut out the introns. So think about it like scissors. And they're going to put the exons back together. So now the exons can go to the ribosome to make the proteins. Okay, so we'll go over this again in class tomorrow, but make sure you try to understand this before you come to class. And so tomorrow in class we can just review it and discuss any questions you may have. And I hope that this was helpful.